This lecture is entitled Introduction to Bioinformatics. The objectives are first to define bioinformatics, to understand why bioinformatics arose as a discipline, and to describe some bioinformatics applications. Let's start by defining bioinformatics. There are many definitions in use, although most have certain elements in common. First, computers are used in some capacity for the analysis of or to model data. Second, the analysis or modeling involves biological data of some sort. It could be genetics data, and it could correspond to humans, but there are many other types of biological data and model organisms or biological systems we could study. Further, bioinformatics doesn't just involve a computer as a passive device to collect or visualize data. Rather, computers are tools for the use of algorithms to gain useful insights and organize and visualize data. Considering all of these issues, the definition of bioinformatics that we have settled on for this project is that bioinformatics is an interdisciplinary field that combines aspects of computer science, mathematics, and statistics to collect, store, manage, analyze, and interpret biological data. Describing the origins may help us put bioinformatics into perspective. A few decades ago, most scientists could look at one biological observation at a time and take notes in a lab notebook to record the observations. However, several technologies started to become available that generate large amounts of biological data that require computers for their analysis. In many cases, the data is captured by a device directly to a computer anyways, so the concept of recording the observations on paper has become obsolete. Gene expression microarrays were developed in the 1990s, and they're one of the technologies that we often use to illustrate how bioinformatics arose. In this figure, what we see here is a gene chip, or a gene expression microarray chip. This square in the middle contains data on over 20,000 genes. A computer actually takes pictures of this area and gets information on what genes are expressed based on its signal intensities. And if we look at that data in a computer, it might look like something like this square on the right. And of course, we need a computer to understand what those signals mean in terms of what genes are expressed and so on. So again, keep in mind that here we're measuring the expression of over 20,000 genes at a time. Looking at the results of so many genes by hand just wouldn't make sense. As we see with technologies such as microarrays, computing has increasingly become necessary for many biological disciplines. And thus, bioinformatics now encompasses many subdisciplines, or sort of many areas where people focus their work. And next, I will discuss some common applications of bioinformatics. Some of these applications we will pursue further along in this course, and others I'm just showing you so that you have a sense of the breadth of bioinformatics and the many possibilities of type of research that can be performed. First, let's discuss genome assembly. And before we get there, though, let's review what the genome is. A genome is the haploid set of chromosomes in a cell. So recall that each cell has a set of chromosomes. For example, this figure shows the set of human chromosomes for a male in a format referred to as a karyotype. We have 22 autosomes, labeled 1 through 22, that we inherit in pairs, as well as sex chromosomes X and Y. And this is how we know that we have a genetic male. In the case of a genetic female, we would have 2 X chromosomes. So the genome is the haploid set of chromosomes, which means that we would take a single copy of 1 through 22 plus X plus Y. And each of these chromosomes is composed of a lot of DNA bases. So here on the right, to remind you what a DNA double helix looks like, it's composed of ACTG bases. And so the idea of figuring out or assembling a genome is to figure out the entire sequence of ACTG bases that make up this karyotype or these chromosomes. In the case of humans, that would result in 3 billion base pairs. So a lot of potential information. Genome assembly is the process of determining the DNA sequences that make up the genome, as I just mentioned. So again, for human chromosomes, what we'd end up with at the end is a whole bunch of DNA sequences like this, sort of like ACTG sequences end to end, which again, in the case of humans, is 3 billion base pairs. So even if we had a machine that could read all 3 billion base pairs at once, we'd actually need a computer just to store that information. In reality, the way we assemble genomes today involves several steps. The process of genome assembly begins in labs and with specific technology. First, we determine what our sample of interest is, let's say a person. Then we obtain a biological sample from that person, which is oftentimes blood, although other tissues that contain DNA are appropriate as well. From said sample, we extract DNA, and we can do this in a lab with various kits that we can purchase. After DNA is extracted, we prepare what's called a sequencing library, and then we use that library on special instruments called sequencers. And this is an image of a specific sequencer by a company called Illumina, but there are other choices of sequencers out there. So what happens after we obtain this sequencing is that a lot of data is generated, and bioinformatics plays a key role. Sequencers actually generate what we call reads, which are little pieces of DNA that represent some portion of the genome. But just by looking at the reads, we don't know where along what chromosome they correspond to. We use computer programs to help us figure that out. So here's an example of the steps involved in assembling a genome in the case that we don't know the genome. We have this sort of reads, these sequenced DNAs. So first we find what reads overlap, and here's just illustrating the example for 2 reads. Portions of them overlap, so we probably think that if we took all the DNA end to end, the reads would go along like that. And we continue to further assemble these reads into so-called contigs and scaffolds, as we see here, until eventually we might have an idea of what an entire chromosome looks like, as in all of the DNA bases that make it up. Alternatively, what we have now, in some cases, is reference genomes, because we already know what the human genome looks like. In those cases, if we're sequencing a new person and we obtain a set of reads, what we would do is try to figure out where along the reference genome these reads map to. So here's just an illustration of showing where some reads might map to along a reference genome. And that can help us computationally speed up this process of figuring out what an individual genome looks like compared to this reference. Next, let's discuss the application of identifying genes in a genome. And this involves comparing genome sequences across species. Doing so can help us to predict functions of similar genes or to find new genes. For example, here's a portion of a gene called ACE2. ACE2 stands for angiotensin converting enzyme, and we're going to look at it across 31 species. So here you see the name of various species. These are common names, and a portion of this gene of ACE2 has been aligned. So you can see that there are certain bases that are almost the same across all organisms and other bases that have differences. By figuring out what are the bases that are exactly the same across species or which ones differ slightly, we can try to figure out how this gene might function a little bit differently in the different species. On the other hand, what we might be interested in is sequencing a new species that's not on this list and we have their genome for the first time. Instead of trying to figure out all of their genes from scratch, what we can do is align their genome and compare it to some of the known species, and then we can maybe more quickly identify if they have the ACE2 gene and if so, where it's located. So this may help us figure out new genes in a species more quickly. Another thing we can do is identify consequences of changing DNA. Comparison of genomes from multiple individuals of one species can help us figure out which sequences result in specific traits, as in observable things, or perhaps other bodily functions, or figure out which changes lead to diseases. So first, we know that most of the genome is similar within species. That's part of the definition of being a species. We also know that some sites differ, but they don't appear to influence any traits. Each of us may have our private changes in our DNA sequence that nobody else has. And then there's other sites that differ across groups of people that maybe we've inherited from our ancestors. But those sites don't seem to be associated with any trait. They don't lead us to have diseases or anything that we can measure. Maybe they do, we just don't know about it. There are other sites that do lead to noticeable changes. And these can, again, be changes in the way we look, in our appearance, our bodily functions, but they can also predispose us to certain diseases. For example, here are some aligned sequences of various tumors from a research study in which scientists were trying to figure out what sequences the tumors had in common. They found that certain motifs that are sort of colored across this figure, certain motifs were in common, as well as some deletions or missing portions of sequences compared to a reference genome, as well as insertions, or sequences that weren't present in the reference. So by finding differences such as these, they can try to figure out a mutational signature for tumors or try to figure out potentially how you might treat this tumor in addition to identifying it. Another application is evolutionary biology. So comparison of genomes across species helps us to understand evolution. And now I'm not talking about looking at a single gene necessarily, but potentially looking at variability across entire genomes. So here's an example of a study about the evolutionary history and adaptation in the African wild dog and 9 other species of canids. So what you see here are images of various canids, some of which are common and you would recognize, such as the gray wolf and the domestic dog. We know, for example, that our domestic dog is most related genetically to a gray wolf than any other. So we think that domestic dogs and gray wolves had a common ancestor. And again, that's partly based on genetic evidence. So what these authors tried to do was to figure out where the African wild dog is in evolutionary history compared to all these other species of canids. So by comparing the genome of them that they had sequenced to these other species, they figured out that maybe there was a common ancestor at this point in evolutionary history in the Pliocene era. So here along the x-axis, you see the different geologic eras. So a very long time ago, there was potentially an ancestor, and one split of it resulted eventually in an African wild dog, and the other split came up with all these other canids eventually over time. So another application is genetic ancestry, and this involves comparing genomes within a species more broadly than at specific locations to better understand ancestral relationships. So it's similar to the idea of the previous slide where we're comparing across evolutionary history, but now within species we might be considering shorter time frames to understand ancestral relationships. For example, we can compare genomes of multiple human populations to determine how we are all related. This figure is from a study called the Human Genome Diversity Panel and consisted of 51 populations. Here they are organized by continent, but a computer program actually produced this figure by clustering the different populations according to similarity across the genome. Another way that we could see how this cluster works is through a tree diagram that looks a little bit similar to the one from the canid example. In this case, though, we're seeing the distance between various human populations. So you can see that Melanesian and Papuan are most similar to one another, and potentially they had a common ancestor over here that diverged into these various other human groups. And you can see at the bottom that the cluster of African populations is most dissimilar to all other human populations, consistent with our understanding that humans originated in Africa and moved out to the rest of the world in a single event, which is the so-called out of Africa hypothesis. So to summarize, bioinformatics is an interdisciplinary field that combines aspects of computer science, mathematics, and statistics to collect, store, manage, analyze, and interpret biological data. There are many bioinformatics applications now because so much biological data is collected by and analyzed with the help of computers.